So for, over a century now, we've basically blamed lactate for that awful burn in our muscles after a hard sprint.
Oh yeah, the classic biological exhaust fume.
Right. We just thought it was waste. You burn glucose, you get energy, and whatever is left over, the lactate just gets pumped out of the tailpipe. But what if I told you that this exhaust is actually like a highly sophisticated, malicious code?
It really is. It completely rewrites the rules.
Exactly. It's a code that cancer cells use to literally rewrite their own operating systems and build this invincible armor against our absolute best therapies. So, welcome to today's deep dive. It is about to completely flip how you view the human body.
Yeah, we are looking at a really paradigm-shifting review from Nature Cell Death and Disease.
We are. It's all about this process called lactylation. And look, whether you're, tracking oncology breakthroughs, or you're a bio nerd, or you're just insanely curious about how life works at a microscopic level, this mechanism changes everything.
It really does change everything we thought we knew about tumor adaptation.
So our mission today is to decode how this supposedly useless byproduct dictates cancer survival, and, more importantly, how scientists are finally learning to hack the system and shut it down. Okay, let's unpack this. Start us at the absolute beginning. How does a simple byproduct from burning sugar manage to, rewrite a cell's software?
What happens through a physical tagging system?
Yeah.
Imagine the DNA inside a cancer cell is this massive library of source code. Lactylation is the process of physically attaching a tiny lactyl tag, which is just a piece of that lactate molecule, directly onto the proteins that manage this code.
And it attaches to specific spots, right? The lysine residues.
Yeah, exactly. The lysine residues, you can picture a lysine residue as like an empty USB port on a protein. But obviously a tag can't just float over and plug itself in.
All right, it needs help.
Exactly. It requires a dedicated, highly coordinated crew of biological machines to do the installation. And we referred to these installing enzymes as the writers.
Okay, so the cell actually has machinery purposefully designed to install this malware. That seems like a massive design flaw in our biology.
Well, it's less of a design flaw and more of a hostile hijacking, really. Let's look at the sheer ingenuity of these writer enzymes. Specifically, there's a pair called AARS1 and ARS2.
Okay, the AARS enzymes.
Right. Now, in a healthy, normal cell, these enzymes have a very mundane day job. They just attach amino acids together to help build standard proteins. But when a cell becomes cancerous, its metabolism just shifts into absolute overdrive, right? It's guzzling glucose and producing massive amounts of lactate.
And that's the trigger.
Yeah. When the lactate levels rise to a certain threshold, the ARS1 enzyme undergoes a physical shapeshift.
Wait, like a structural transformation?
Precisely. It undergoes a conformational change, and when it changes shape, it physically exposes this hidden sequence on its surface called a nuclear localization signal.
Oh, wow.
Think of this signal as like an all-access VIP badge. So suddenly this mundane worker protein flashes its badge, gains access to the cell's nucleus, the secure server room where the DNA is kept, and it completely changes jobs.
It becomes a lactyltransferase.
Exactly. Its sole purpose is now to plug lactyl tags into nuclear proteins.
That is wild. It's literally like a barista suddenly pulling out a Pentagon badge and just walking into a classified briefing to rewrite national security protocols.
That is actually a remarkably accurate way to picture it. And they take it a step further with something called a metabolon.
Right, the VIP pipeline.
Yeah. Rather than synthesizing these tags and just, throwing them out into the crowded cell, hoping they find a USB port, the cancer cell builds a private internal pipeline.
So it's hyper efficient.
Incredibly. Two enzymes that create the tag, ACSS2 and GTPSCS, they physically link up with the writer enzyme that installs the tag, which is KAT2A. So the tag is created and passed internally, hand-to-hand, directly to the DNA.
It completely bypasses all the chaos of the rest of the cell.
Right. It avoids global scarcity entirely.
Okay, wait. I'm stuck on something here, though. I was looking through the notes on the enzymes involved, and it mentions HDACs. Aren't histone deacetylases normally the erasers of the cell? I thought their whole job was to remove tags, not write them. Am I misunderstanding how they work?
No, you aren't misunderstanding at all. You've actually just hit on one of the most brilliant and paradoxical mechanisms in this entire review.
Okay, thank God. I thought I was losing my mind.
No, you are completely right. In a normal environment, HDACs are erasers, but they are what we call context-dependent bifunctional enzymes.
Context-dependent, meaning the environment dictates the job.
Exactly. Everything changes based on the concentration gradient of lactate. In a normal cell, lactate levels are low, but inside a tumor, lactate spikes above 0.5 millimolar. That intense concentration physically forces the HDAC enzyme to run in reverse.
Oh my God. So the high concentration of the exhaust fume literally forces the cellular eraser to become a writer. It turns the antivirus software into a virus.
Exactly. And, it doesn't even always need enzymes to pull this off.
Wait, really? It can happen without enzymes?
Yeah. The volatile chemical environment of a cancer cell produces these other glycolytic byproducts, like MGO and LGSH. These chemicals can force a lactyl tag onto a protein completely non-enzymatically.
Just by chemical force.
Yep. For example, LGSH will utilize nearby cysteine residues. So other amino acids sitting close to the target to act as like a chemical wingman, guiding the lactyl group right into the lysine port. This creates a very specific tag known as KD-lay.
Wow. And I know the review mentions there are dedicated erasers too, right? Like HDAC 1 through 3 prefer removing D-lactyl marks, and RRT 1 through 3 prefer L-lactyl marks.
Yes. The cell still tries to regulate it, but the writers are just overwhelming the system.
Okay, we need to lock in this first concept for you listening at home. We're going to do these quick checkpoints as we go to keep all the pieces organized because this is dense. So first, lactate isn't waste. It is the raw material, the malicious code. Second, the cancer cell hijacks normal enzymes, like the moonlighting ARS1 or the reversed HDACs, using them as writers to install this code.
Yep, the construction crew.
Third, even without those enzymes, the toxic chemical environment can just force the installation of these tags anyway. So here is the interactive prompt, the question we need to answer next. Now that the cancer cell's proteins are covered in these lactate tags, these little USB drives full of malware, how does the cell actually read the code and change its behavior?
Well, that transition from installing to executing the code requires reader proteins. A lactyl tag by itself, it doesn't really do anything. It's just a physical bump on a protein.
Right, it's just sitting there.
Exactly. To execute the malware, reader proteins have to scan the genome, recognize the specific shape of the lactyl tag, and translate it into a biological action.
But how exactly does a protein recognize a tag? It doesn't have eyes.
It comes down to structural lock and key mechanics. Take a reader protein called BRG1 or another one called DPF2.
EPF2, right?
Yeah. DPF2 has this specialized area called a hydrophobic pocket. It is physically shaped to perfectly envelop one specific lactyl tag on the DNA spool, known as H3K14-OLA.
So it docks onto it like a puzzle piece.
Exactly. And once DPF2 docks onto that tag, it changes the local DNA structure, physically unspooling it to expose oncogenes, which are the genes that cause uncontrolled growth. It's essentially using the tag as a foothold to reach over and hit the gas pedal on tumor expansion.
That is fascinating. And there are others too, right? Like TRM33.
Yes, TRM33 uses a totally different structural key called a PhD bromodomain to read the tags.
Okay, PhD bromodomain.
Right. But what TRM33 does is wild. It doesn't just hit the gas pedal for growth. It specifically reads tags that send signals outward to manipulate macrophages, which are our body's immune cells.
Oh, wow. So it's doing external crowd control.
Exactly. We'll get into that immune manipulation shortly. But the core concept is that these readers execute different commands based on which tag they find.
Okay, here's where it gets really interesting. I want to look at what happens when the tag isn't just a signal, but a literal physical obstruction. Because the review mentions lactylation can cause a, quote, structural metamorphosis of non-histone proteins, like P53.
Yes, P53.
Right. Most people who follow biology know P53 as the guardian of the genome. It's our primary tumor suppressor. So how does lactylation deal with P53?
It neutralizes it entirely through a mechanism called steric hindrance.
Steric hindrance.
Yeah. Normally, P53 works by sliding into a very specific groove in the DNA to stop a damaged cell from multiplying. But the cancer cell uses lactylation to attach these bulky tags directly onto P53 at two precise locations, K120 and K139.
Steric hindrance. So that basically means two objects cannot occupy the same space at the same time.
That's it, exactly. By adding this bulky, negatively charged tag right where P53 needs to grip the DNA, it physically prevents the protein from fitting into the slot. The tumor effectively corrupts the system's emergency brake so it can never be pulled.
That is terrifyingly simple. It just makes the brake pedal too fat to press.
Yep. But it can also do the exact opposite, right? It can supercharge the programs the cancer actually wants to run.
Yes, it can. Let's look at a DNA repair protein called XLF. When XLS is lactylated at position K288, the tag fundamentally alters the protein's electrical charge.
Like an electrostatic shift.
Yes, it creates this electrostatic magnetic boost. This allows XLF to snap together with another repair protein, CoA80, at just incredible speeds. It vastly accelerates the cancer cell's ability to repair its own DNA.
Oh, and doesn't it also work with other modifications, like PTM crosstalk?
Yeah, lactylation frequently cooperates with other modifications. For instance, in T-cell leukemia, it synergizes with an acetylation mark called H3K27XE to just hyperactivate oncogenes. It's a highly collaborative network.
Yeah, so it breaks the brakes and supercharges the repairs. Let's do our second checkpoint to keep this organized for everyone listening. First, the newly installed lactyl tags are executed by reader proteins like DPF2, which use specialized structural pockets to lock onto the tags and turn on cancer-causing genes.
Perfect. Second, lactylation can physically break vital proteins, like adding a bulky tag to P53 so it can no longer fit into the DNA to stop the tumor. And third, it can magnetically supercharge the bad proteins, like XLF, accelerating tumor survival. It competes or cooperates with other modifications to get this done.
Right.
Which brings us to a massive prompt for you to think about. If tumors can supercharge their own DNA repair systems like a hyperactive mechanic, what happens when we try to destroy them with targeted DNA-damaging chemotherapy?
Well, this is where we see the phenomena of intrinsic resistance. The tumor uses this lactylation software to literally armor itself from the inside out.
Because the whole goal of radiation or traditional chemo is to cause DNA damage, right?
Exactly. The goal is to inflict so much catastrophic DNA damage on the cancer cell that it undergoes apoptosis, which is programmed cell suicide.
But the cancer cell is anticipating that.
It is actively countering it. We mentioned XLF, but lactylation also targets other emergency first responder proteins like NBS1 at a site called K388 and BLM at K24.
NBS1 and BLM.
Right. And the moment the chemotherapy shears the DNA, these lactylated proteins swarm the site and instantly patch the holes. I mean, the drugs simply cannot outpace the lactylation-fueled repair rate.
It's like trying to sink a ship with a cannon, but the crew has an army of super speed welding torches. But it's not just DNA damage they survive, right? I was reading this section on ferroptosis, and it absolutely blew my mind.
Oh, the iron starvation tactic.
Yeah. For those listening, ferroptosis is literally death by iron. When iron interacts with cellular fats, it causes a toxic rusting chain reaction that just shreds the cell. So how does the cancer survive that?
The mechanism here is just brilliant. To survive iron death, the tumor obviously has to prevent iron from getting inside. So it lactylates a regulatory protein called LSD1.
Okay, LSD1 gets the tag.
Right. And once tagged, LSD1 shuts down the production of the TFRC receptor. TFRC is essentially the loading dock door that lets iron into the cell. By eliminating that door, the tumor pulls up the drawbridge.
It deletes the loading dock so the iron payload can't be delivered.
Exactly. It starves itself of iron to deny the chemotherapy the very weapon it needs.
And doesn't it also build up a defense for the iron that's already inside?
Yes, simultaneously, lactylation triggers the massive production of glutathione, or GSH, which is a very powerful antioxidant. So it locks the doors to new iron and deploys neutralizers to clean up the toxic rust from whatever iron is left inside. It covers all bases.
I did notice a strange paradox with copper-induced death, though, ferroptosis. The review called it a duality.
Right, because lactylation is a precise software, not a blunt instrument. If a protein called NUDT21 receives a lactyl tag, specifically at K23, the cell becomes highly resistant to copper-induced death.
Okay.
But if a different protein, MENTL16, gets tagged at K229, the cell actually becomes hypersensitive and dies from copper. It highlights how incredibly specific this coding language is.
Wow. Which honestly brings me to the craziest survival mechanism in this whole paper. The diapause-like cancer cells, or DLCCs. When I read this, I genuinely had to pause. Walk us through what happens when a cancer cell realizes it's losing the battle against chemotherapy.
This is wild. When the stress becomes too great, a specific gene called SMC4 shuts down. This triggers the cancer cell to enter diapause, a state of deep cellular hibernation.
It just completely stops dividing entirely.
Exactly. To an oncologist looking at a scan, the tumor looks dormant, maybe even dead. But internally, it's an active hibernation. The cell ramps up glycolysis, pours out lactate, and uses that lactate to heavily tag its histones.
So it reboots in safe mode. And what is it doing while it's hiding in safe mode?
It is using those new lactyl tags to construct ABC drug efflux pumps. specifically models ABCC2, 3, and 10.
Efflux pumps.
Yeah. These pumps are literal physical bouncers embedded in the cell wall. When the cell finally wakes up from hibernation, any chemotherapy drug that tries to enter is immediately pumped right back out into the bloodstream.
It literally uses the downtime to build a firewall that actively deletes the drugs.
Man. Okay, let's lock in this third checkpoint on internal resistance. First, lactylation supercharges multiple DNA repair pathways to instantly patch the holes made by chemo.
Second, it actively rewires iron and copper metabolism to survive targeted attacks, like deleting the iron-loading dock to prevent ferroptosis. Third, under intense stress, it enters a hibernation state to secretly build efflux pumps that will spit toxic drugs back out.
Spot on.
But here's a thought for you to chew on if you're listening. We've seen how the tumor protects its inside. But a tumor doesn't exist in a vacuum, right? It is sitting inside a human body, surrounded by our immune system. So how do you think it handles external threats?
It handles the external threat through extrinsic resistance, which is essentially cellular psychological warfare.
Psychological warfare?
Yeah, the tumor doesn't just hide, it actively brainwashes the microenvironment around it.
Let's start with the hiding part, because the stealth tech is fascinating. PD-L1.
Right. PD-L1 is a natural protein found on the surface of healthy cells. It's basically an ID card that tells the immune system, hey, I belong here, do not attack. Cancers often hijack this to create an invisibility cloak.
And lactylation helps with that.
What this review proves is that lactylation is the tailor making the cloak. In colorectal cancer, a lactyl tag physically stabilizes the PD-L1 protein post-translationally, so it never degrades.
Oh, wow.
And in leukemia, a lactyl tag called H4K-Fiavola is placed right on the gene's promoter, epigenetically forcing the cell to constantly manufacture more of these fake ID cards.
Okay, so it's practically invisible to the immune system, but you mentioned brainwashing earlier. How does it reprogram the immune cells that do manage to find it?
This is where the tumor weaponizes its own exhaust. The cancer cell constantly pumps raw lactate out into the surrounding tissue.
Okay.
Our own immune cells, specifically macrophages, absorb this lactate. And once inside the macrophage, the tumor-derived lactate forces a specific modification called H3K80
Wait. So the tumor is literally dumping its metabolic waste onto our immune cells, and that waste contains a code that installs malware on our cells.
Exactly. Our macrophages normally exist in an M1 state, which is aggressive and tumor-killing. But that H3K80 tag physically rewires the macrophage, flipping it into an M2 state.
And M2 is bad for us.
Right. An M2 macrophage acts as a healer and protector. The tumor has literally rewritten our own antivirus software to act as its personal bodyguard.
That is just diabolical. And it expands other protective cells too, right? Like TREGs.
Yes. It lactylates a protein called APOC2 at K70 to rapidly expand regulatory T cells, or TREGs, whose entire job is to suppress immune responses.
It's shutting down the whole neighborhood.
It is. And for the heavy hitters of our immune system, the natural killer, or NK cells, the tumor's lactate environment tags a protein inside the NK cell called ROCK1, specifically at K73.
ROCK1.
Right. That specific tag physically shatters the mitochondria inside the NK cell in acute myeloid leukemia, completely destroying its energy source.
Yeah.
It leaves our best killers paralyzed.
Oh my God. It builds an invisibility cloak, it brainwashes the local police to protect it, and it cuts the power to the military.
Exactly.
And if that wasn't enough, it uses lactylation to build its own supply lines through angiogenesis, right?
Oh yeah. A tumor needs massive amounts of blood to survive. By lactylating specific transcription factors like HIF1A and histones like ASH2L at K312, the tumor triggers the release of a signal called VGGFA.
VGFA?
Yeah, and this signal forces the human body to sprout brand new blood vessels, custom piping nutrients directly into the tumor core.
Man, okay, let's summarize this terrifying extrinsic network before we talk about how to break it, because I need some good news.
Yes.
First, lactylation forces the expression of PD-L1, hiding the tumor from the immune system.
Yes.
Second, it physically brainwashes macrophages and TREGs to protect the tumor while disabling the NK killer cells. Third, it forces the body to build custom blood vessels to feed the cancer. So I'm listening to this, and this network seems completely invincible. But if lactate is the key to the whole operation, what happens if we just cut off the supply?
That is the exact question driving the new era of clinical translation. If lactylation is the operating system, keeping the cancer alive, we have three distinct strategies being developed right now to crash that system: metabolic intervention, epigenetic reprogramming, and precision site-specific inhibition.
Let's start with metabolic intervention, which is basically starving the beast. The review highlights an FDA-approved epilepsy drug called stiripentol. How does an anti-seizure medication fight cancer?
Well, it's all about repurposing the mechanism. Stiripentol happens to be an incredible inhibitor of LDHA, which is the core enzyme responsible for producing lactate in the first place.
Ah.
If you give this to a tumor, you cut off the global lactate supply. You strip all the raw materials away from the writer enzymes. In preclinical models of gastric cancer and glioblastoma, treating the tumors with stiripentol completely reversed their resistance to standard chemotherapy. Without the lactate, the armor just falls apart.
That's a massive win for drug repurposing. But what about the second strategy, epigenetic reprogramming? How do we jam the machinery if the lactate is already there?
We use decoys.
Decoys.
Yeah, for example, a simple molecule like beta-alanine physically looks enough like lactate that it can compete for the binding site on the ARS1 writer enzyme. If beta-alanine is sitting in the enzyme's chair, the lactate can't get in, and the tagging process halts.
Oh, that's clever.
Right. Scientists are also designing custom fusion peptides, synthetic proteins, that wedge themselves between ACSS2 and KT2A, physically breaking that private underground pipeline we talked about earlier.
Okay, but for a listener who wants to know what this means for human beings right now, are we just curing cancer in petri dishes and mice?
No, we are already in human trials with the third strategy, precision site-specific inhibition.
Okay, lay it on us.
Instead of shutting down all lactate, which, normal cells do need in small amounts, we target the specific pathological tags. Remember the BLM protein? The one that supercharges DNA repair when it gets a lactyl tag at K24?
Yes, the super speed welding torch.
Exactly. Researchers discovered that an existing drug called irinotecan specifically binds to BLM and physically blocks that exact K24 lactylation site. They took this into a phase 1 clinical trial.
I read the results of this trial in the source material, and it is staggering. Walk us through what happened.
They tested it on 6 human patients with recurrent anthracycline-resistant bladder cancer.
So highly resistant.
Extremely. These are patients whose tumors had completely learned to ignore our standard, highly toxic chemotherapy. The tumors were heavily armored. But by adding irinotecan to the regimen, specifically to block that one single lactylation site on the BLM protein, the armor was breached. Out of those 6 patients, 100% were recurrence-free at the 6-month mark.
Wait, 100% recurrence-free for a cancer that was previously ignoring the chemo?
100%.
And with only mild, like grade 1 or 2 adverse side effects?
Exactly. It is a profound proof of concept.
So what does this all mean?
It means that if we can map what we call the lactylome, the specific pattern of lactyl tags a patient's tumor is using to survive, we can predict exactly which therapy it will resist and deploy a precision drug to rip that specific shield away. It proves metabolic signals dynamically encode cancer vulnerabilities.
Let's do our final recap of how we fight back, because we are on the verge of clinical application here. First, we can use old drugs like stiripentol to cut off the lactate supply.
Right.
Second, we can design new synthetic decoys to jam the writers from attaching the lactate. Third, precision drugs like irinotecan are already showing incredible 100% recurrence-free results in early small human trials by targeting specific lactylation sites. We are looking at a complete revolution in oncology. This isn't just about killing cells anymore. It's about rewriting their software.
It really is.
And that leaves me with one final mind-bending thought for you to take away from this deep dive. If a basic metabolic byproduct like lactate, something we have known about for over a century and completely dismissed as just the exhaust fume of a hard workout, can actually enter the nucleus of our cells, fundamentally rewrite our epigenetics, and dictate whether a deadly tumor lives or dies. What other everyday metabolites are secretly pulling the strings?
It's a huge question.
What other exhaust fumes generated by our daily diet, our stress hormones, or our environment are acting as the hidden software running human aging, Alzheimer's, or heart disease? We have spent the last 100 years thinking we were just staring at the exhaust pipe of the human body. But it turns out we may have been looking at the steering wheel all along. Thank you for taking this deep dive with us today. Keep questioning the information you consume and keep wondering about the incredible machinery operating inside you right now. We will catch you on the next one.